Hey everyone. Today we are starting a new topic, biotechnology in the pharmaceutical industry. Again, very relevant to what's going on in the world today. If you recall, we finished the end of the microbiology unit talking about vaccines, um, which kind of tied up our discussion of viruses. Vaccines are obviously tied to the pharmaceutical industry. So these kind of go hand in hand, which is why we're going to do this topic next. Um, so number one, it's relevant to what's going on, but number two, it also ties into what we just finished talking about, which are vaccines. Okay, so to break it down, biotechnology is starting to become an important part of the pharmaceutical industry. Um, for hundreds of years, medicine has revolved around chemical treatments, surgery, things like radiation. I mean, if you want to go way back, it goes back to, lobotomies and bloodletting. Um, Biotech has always been a very small piece. You could almost consider homeopathic medicine to be biotech, using herbs and essential oils and um, spices and diet, things like that to treat diseases. What we're talking about here is much more manipulative, uh, somehow editing or changing or engineering a living thing to be used in medicine. So we're moving away from, chemical drugs and surgery and trying to fix diseases using biological materials instead. So, one component of biotechnology in the pharmaceutical industry is the manufacturing of drugs. The typical drug in the pharmaceutical industry is a small molecule, a chemical. A good example is aspirin. Think of your ibuprofen, your what else? Of course, I literally can't think of any other medicine right now, but you get the idea. Your over-the-counter small molecule tablet, right? You're consuming some kind of chemical. Biologics are where we get into the biotech industry, using actual biological molecules as medicine. Instead of just a chemical made up of atoms, we're using a biological molecule. Maybe, an antibody or a protein, something like that. So there are small biologics and large biologics. And just to give you a reference point, so the pharmaceutical industry makes an obscene amount of money every year. And this is an old graphic by now. When this data was compiled, $700 billion of the pharmaceutical industry came from traditional methods, and $175 billion came from biotech. So it's a small proportion, but it's growing. Just to give you an example, these are some of the giants of big pharma, and this data shows you how much of their revenue has changed to become biotech. So this is a change in percent of revenue from biopharma. So the change in percentage. The biggest change would be Sanofi Aventis, right? 53% of their revenue changed to be coming from biotech. One you might be familiar with is Bristol Myers Squibb. There's a location pretty close to South Brunswick Obviously, you probably know Johnson& Johnson. Bayer is a big one. So you can see that some are devoting more than others, but many of these big pharma companies are investing in biotech. So again, the biggest part of biopharmaceuticals, using biotech in the pharmaceutical industry, comes from those biologics, those medicines that are made up of some kind of living material, usually a protein. They can be extracted directly from the living system, or they can be manufactured using bacteria. So let's say you wanted to treat a chronic condition using a protein. You could either harvest that protein from a person, who makes it, or you could kind of copy that gene into the bacteria, and then they will make that protein for you. So there are several different types of biologics. The first type of biologic is when the medicine, the molecule itself, is nearly identical to the body's own proteins. So it's not really engineered in any way. You're giving somebody a protein. Probably the most common example is insulin for diabetics. Their body does not produce insulin, so you just need to give them insulin. You don't have to change it or modify it or engineer the protein in any way. They're just missing insulin. So their medicine is the protein, right? It exactly mimics the body's own proteins because it is the body's proteins. It's just coming from another source because they can't make it themselves. Some other examples are erythropoietin. This is the protein that stimulates blood cell production. So if some people don't have enough of this, you give it to them as a medicine. So again, insulin is probably the most common example of that first type of biologic. More often than not, it is produced by bacteria, E. coli actually. The first successful extraction of insulin from bacteria occurred in 1978 at a very brand new biotech company called Genentech. Today, Genentech is one of the, big names in biotechnology, and this research really put them on the map. They spliced the gene for human insulin into bacteria. The bacteria transcribe and translate that gene just as their own DNA, and they produce the protein in high quantity, and we extract it and put it in a bottle and sell it as medicine. So the crazy thing about insulin is that it has not changed since 1978, and yet the price just keeps going up and up and up every year. If you're interested in the pharmaceutical industry, that's a really interesting thing to read about. The exploitation of insulin. Somebody's making money off of a naturally occurring human protein, made by everyone's pancreas. So something to think about and something to look into if you're interested. So this is how they make it. They take it from a healthy pancreas cell. They take that gene, right? Here's the nucleus, here's the DNA. They extract the gene for producing insulin, and they stick that gene into a plasmid. Remember, the plasmids are the circular pieces of DNA that some bacteria have. It gives them special abilities. So that little piece of DNA gets spliced into the plasmid. That plasmid gets put into the bacteria. The bacteria reproduce. The protein that gets made is extracted and put into a bottle and sold to diabetics as a treatment. Another example is hemophilia. Hemophilia is a genetic disease where the affected people cannot produce blood clotting factors. So normally when you get a cut and you're bleeding, a normal person produces these blood clotting factors that then help form a platelet plug and stop the bleeding. In people with hemophilia, they have no clotting factor or less clotting factor and form a weak platelet plug that does not stop the bleeding, so bleeding continues. It is a very dangerous disease. If you get a cut, it does not stop bleeding. There's no clotting. And this clotting factor is called factor 9, or sometimes it's abbreviated to FIX, factor Roman numeral 9. And a biologic that biotech companies have produced for hemophilia is called Benefix. So the way they make it, they take the human gene that codes for FIX, blood clotting factor 9, and they transfer it to Chinese hamster ovary cells. I looked it up once. I don't know why they use Chinese hamster ovary cells over bacteria. I do know that Chinese hamster ovary cells, so literally the cells from the ovary of a female Chinese hamster, reproduce rapidly, in a test tube, in a petri dish. I'm assuming that the blood clotting factor is a very complex protein that perhaps does not fold correctly in bacteria, so it needs to be in a mammal cell, a eukaryotic cell for that matter, and the CHO cells, the Chinese hamster ovary cells, reproduce rapidly enough, at a similar or comparable rate to E. coli and make the protein better. So they're used because they can produce the recombinant protein on a large scale. I just can never find information on why they don't just use E. coli, which also produces a high number of proteins. I'm assuming it is for the quality of protein product. So the Chinese hamster ovary cells wind up transcribing and translating that gene just like their other genes, and they produce the protein. That protein is harvested from the CHO cells, purified, and sold as Benefix. So this is literally just human blood clotting factor 9. It's a protein that gets injected into people with hemophilia. That way, if they get a cut, they have that blood clotting factor and their bleeding will stop. So another type of biologic is called a monoclonal antibody. These are similar to the antibodies that the human immune system uses to fight off bacteria and viruses, but they are custom designed to bind much more specifically to their target molecule. So when we learned about vaccines, a bunch of the articles given to you talked about how vaccines actually work, and talked about the immune system. So the human body produces antibodies, which are proteins in response to some type of illness. It is the immune response. Antibodies, what they do is they attack and kill foreign invaders. Could be like allergens, dust, pollen, peanuts, all the way up to viruses and bacteria. So antibodies are what your body produces to fight off invaders. Antibodies bind to and attach to their target and somehow render them inactive. In the case of a monoclonal antibody, they are much more specific. So regular old antibodies are already pretty specific, right? There's only one antibody for the coronavirus. There's only one antibody for pollen, things like that. One antibody binds to one antigen, it's called, the thing they bind to. But monoclonal antibodies are even more specific because they are custom designed in a lab. They are engineered by humans. A classic example is Humira. You might have heard of Humira in commercials, those drug commercials. It is a monoclonal antibody that targets and inhibits tumor necrosis factor alpha, TNF alpha. This results in an anti-inflammatory response. So it inhibits this protein. And this is an anti-inflammatory response. So Humira is prescribed for those with arthritis, Crohn's disease, and psoriasis. And again, it's a monoclonal antibody. So it's a very specific antibody that binds to TNF alpha and inactivates it. So just to show you, arthritis is one of the most common diseases associated with Humira. It's prescribed for it. So, for example, in your knee, you have cartilage right here that protects the joint from rubbing up against each other. TNF alpha, that protein, what it does, it actually breaks down your joint tissue. It's caused by an overactive immune system. So your immune cells release this TNF and it degrades the cartilage on your knees, which really results in inflammation and pain because your bones are rubbing right up against each other. What Humira does is it binds to this TNF and makes it inactive so that it can't break down that joint tissue. Okay? Another type of monoclonal antibody is used to treat breast cancer. So this would be a breast cancer cell with these HER2, I think they're receptors, yes, receptors on the surface of the cell. Okay, so this would be a normal cell. It's misleading. This is breast cancer cell. So this is just a breast cell. It's normal, right? It's got the right number of HER2 receptors, and those receptors send signals telling cells to grow and divide. So if there are too many HER2 receptors, then they're going to be giving too many frequent signals to divide. So the cell divides out of control, which is what causes that breast cancer. So Herceptin is a monoclonal antibody biologic that binds to those HER2 receptors and it activates them again, decreasing the signal that gets sent to the breast cancer cell and inactivating the receptor from doing its job. So again, it's a specific antibody that binds to this protein, makes it inactive, and prevents breast cancer. The third type of biologic is a chimeric protein or fusion protein. If you've ever studied Greek mythology, then you've probably heard of a chimera. A chimera is a living thing that is made up of two or more animals. Think of a centaur, half human, half horse. That would be a chimera. So chimeric proteins are, again, scientifically engineered proteins made in a lab that contain two different proteins. It's like a Frankenstein protein. You take a little bit from this protein and a little bit from this protein and fuse them together, and now it'll have two functions in one protein. So again, a great example is a tanocept. Forget the common name for a tanocept. It'll be on one of the next slides. So again, this is a TNF alpha blocker that is even more highly specific than the monoclonal antibody. So it contains two pieces. It has a TNF receptor, so something that regularly binds to the TNF alpha, and it has an antibody segment. So it's part like monoclonal antibody. This is the antibody part, and then it also has a receptor for TNF. so two pieces in one protein. So the receptor piece provides specificity as well as the antibody piece. So they both work together in this one protein to be really, really highly specific to bind to TNF alpha and block it from breaking down the cartilage, preventing arthritis and all that. So it's a similar drug, Enbrel, that's what it's called. So Enbrel is a tanacept. So again, this would be a normal amount of TNF, regular cartilage. Too much TNF breaks down the cartilage and causes the bones to rub together and breaks them down. Enbrel, you can see if you look really close, there's that orange piece and the pink piece, right? It's a chimeric protein. It's got two parts. It's got the antibody part and the receptor part. So it binds to that TNF, makes it inactive, and prevents any further damage. It cannot reverse damage, but it can prevent any further damage. Okay? So those are your three types of biologics. Oh, I have more pictures. So this one would be your Enbrel, right? It's got two pieces. It's chimeric. This one right here is Humira. This is the long scientific name, adalimumab. Yeah, there you go. This is Humira, and then this is your TNF, the thing you want to break down and inhibit. So again, chimeric has two different pieces on it. This would be your monoclonal antibody. Okay, specific, highly specific, even more highly specific. Okay, and they're actually showing you right here is that receptor. This is the TNFR. This is what usually captures that TNF. So a little piece of this is engineered to be part of this medicine so that it perfectly binds to that TNF. Okay? And again, another picture of it. Okay? Chimeric. This one actually has, I don't know what these are. Oh, that's immunoglobulin. So a little piece of antibody and a little bit of mouse variable region. So for some reason, I'm assuming it's just a lab animal used to make this chimeric protein. Chimeric protein, two pieces, and then this one would be monoclonal because it's made up of it's just one protein, right? This is a monoclonal antibody. This would be chimeric because it has two different parts to it, right? This is the antibody, and then it was engineered to contain this other piece as well. Okay? Okay. Just a little bit of background. So biologics are really expensive. Biologics, it seems like a simple process, right? You're just taking proteins that already exist and perhaps engineering them a little bit. But, if you think about insulin, you don't really have to do anything to it. You don't have to manipulate it. You just have to get it and deliver it to the people who need it. But biologics are inherently very expensive. The fact that it's living material, it's not just a simple chemical. It's a living molecule that takes the expensive part of it is isolating and purifying the proteins. So you can grow that insulin in the bacteria, but then you have to extract it from the bacteria. That's the expensive part, and purifying it, making sure you have only insulin and not any of the other pieces of the bacteria. So biologics get a pharmaceutical company that releases a biologic gets 20 years of a patent, basically. After 20 years, the patent expires and biosimilars get made. This is like your generic. So all pharmaceutical companies do this. When they release a new drug, they have a certain time where they have the patent and nobody else can make that drug. That's where they make all their money. And then after 20 years, all pharmaceutical companies have a chance to make and sell a similar drug. In biotech, those drugs are called biologics and then biosimilars. So you might be familiar with the word generic. I just want to show you how much Humira costs. So Humira is still a biologic. It has not reached its 20-year biosimilar yet. So if you had arthritis and needed to buy Humira over the counter, look how it's like this is ShopRite. $5,489. Stop and Shop. And I don't even know how long, like, how much of a dose it is. One carton, 2 pens. I don't know how long that would last you. That's just crazy, and that's with that coupon. So, and that's again for Humira. I wonder if I can find insulin. So, not as bad. But yeah, those biologics, a lot of research and development goes into those, which is why they're made, they're so expensive. So something to think about, again, the pharmaceutical industry. So how are these antibodies produced for use in the biotech industry? We've seen what they are and how they work, and the different types, 3 different types of biologics. But how are these antibodies produced, or at least extracted, for the biotech industry? So there's a couple types of antibodies. Polyclonal antibodies are one type. So we talked about monoclonal. Polyclonal antibodies. So I feel like I'm missing a slide here. Maybe that, yeah. I think because, yeah, those slides were with my vaccine stuff. So let me just give you some background. Again, you know what? Let me actually try to go find those slides. Okay, wasn't too far. So a little rewind, because we didn't get to go in depth when I did the vaccines. So antibodies are proteins that specifically bind to an antigen. So the antigen is the foreign substance, and the antibody is the protein produced by your immune system to attack the foreign substance. So antibodies are made by your cells to attack foreign invaders. The antibodies are your immune system. Okay? So once activated by the infection of a pathogen or any type of antigen, antibodies stay in your bloodstream and continue to provide resistance against that pathogen. Okay? So this is how vaccines work. Once you get exposed to an antigen in a little teeny tiny dose, your body will produce antibodies, which then stay in your bloodstream and protect you should you ever come in contact with that antigen. So if a COVID-19 vaccine is made, you will, you get the vaccine. It will contain a little bit of the virus. It'll be a very weakened form of the virus. Your cells, your body, will produce antibodies that attack the COVID-19 antigen, and then you'll have protection against the virus, right? So you expose yourself to a little bit of the virus so that if you ever become exposed to the real virus, your immune system is prepared. So antibodies are produced by B lymphocytes, B cells, to attack and kill toxins. So these are the cells that produce the antibodies. Remember, antibodies are proteins. So these cells produce the antibodies. Antibodies recognize and bind to the specific epitopes on the surface of the antigen. These are, again, proteins that stick out of the surface of the antigen. So if this is the antigen, right, the bacterial cell, then these green shapes are the epitopes. And the antibodies bind specifically to the epitopes. So the antibody doesn't bind to the entire bacterial cell, it binds to these receptors called epitopes on the surface of the antigen. So here's your antigen, these are your epitopes, okay? So they work by either labeling the antigen so that your other cells in your immune system can find them and kill them. They could kill the target directly, or they could, I think that word, immobilize. So antibodies work by either labeling the antigen for destruction, killing directly, or immobilizing, making it so that they can't move and do their function. And then another thing about antibodies. So there are primary antibodies, which are proteins that bind specifically to antigens. But then there are also secondary antibodies that bind to primary antibodies. This becomes important in something we'll talk about another time called the ELISA test, but just keep in mind that there are primary antibodies and secondary antibodies, not to be confused with monoclonal antibodies and polyclonal antibodies, which we'll talk about in just a little bit. But primary antibodies bind directly to the antigen, while secondary antibodies bind to primary antibodies. Just a little background about antibodies and antigens, something you might have learned about in another bio class or blood types. So these are the 4 blood types, A, B, and O. The negative and positive is something else, but these are the 4 major blood groups. A person who is blood type A has A antigens on their red blood cells. So you have proteins that stick out of your red blood cells called antigens. Well, they'd actually be the epitopes, but you have the A antigen and the B antibody. This is why if a person who is blood type A is given blood type B by accident, those blood cells will actually be attacked by our immune system because you have anti-B antibodies. You can't have anti-A antibodies if you're type A because you'll just attack your own cells. So you have the A antigen and the B antibodies. Somebody who is blood type B has B antigens and anti-A antibodies. So they attack anything with A antigens. They won't attack their own cells, but they'll attack type A blood cells. A very rare blood type is group AB. You have both A and B antigens, meaning you can't have any antibodies. You have no antibodies in your bloodstream to attack red blood cell antigens, which makes group AB the universal acceptor. Anyone who is type AB can accept blood from anyone because they have no antibodies in their system to attack blood cells. Type O is the most common type of blood. You have no antigens, but you have all antibodies. This means that type O is the universal donor. They can give their red blood cells to anyone because there are no antigens. Even a type B person would not attack this type of red blood cell because there are no antigens. However, type O can only receive blood from other type O people because they have both of the antibodies. Okay, so just a little bit of background for you. Okay, that was something we missed from the vaccine lesson that I usually give. So now I'll go back. Okay, so again, that's all about antibodies, how they work in the immune system. Now going back to the pharmaceutical industry, we have to talk about how they're made in the production of medicine. So polyclonal antibodies are made by injecting the antigen into a host. That host, their B cells, their B lymphocyte cells, will produce antibodies to attack that antigen. So you can see this B cell is producing an antibody that binds specifically to this epitope on the antigen. This B cell is also producing an antibody that is specific to this epitope on the antigen. So now you have what are called memory B cells. These B cells now have all of these antibodies that will forever know to attack this antigen. You then extract the blood from the host, knowing that it will have all of those antibodies made by the B cells. So this blood serum will have, it's called the antiserum. It contains the antibodies to attack this antigen. I don't know if you've ever seen the movie Outbreak. It's shown in health class a lot. They keep talking about the monkey that they have to find because it is it's the host, right? So its blood contains the antibodies needed to kill the antigen. So the virus is spreading around the town and it's killing people so quickly that their immune systems don't even have time to react. Their immune systems can't produce antibodies. So nobody is producing antibodies except for that very original host. So they have to find that monkey and extract the blood from it. So, and they call it the antiserum in the whole movie. It's actually very scientifically accurate. And if they find that monkey, then they'll have the antibodies needed to attack the antigen. So they can just inject that antibody into the patients and it will theoretically cure the virus. Of course, it has to be compatible cross species, but that's a whole different story. So yeah, if you want to make a polyclonal antibody, you inject the antigen into the rabbit. The rabbit B cells make the antigen, and then make the antibodies to attack the antigen, and then you extract the blood from the rabbit, and that blood will be full of the antibodies. Now, monoclonal antibodies are a little bit different. What makes this polyclonal is that these antibodies what makes this polyclonal is that every B lymphocyte cell in the rabbit is making antibodies. So you have a bunch of different cells here all making their own antibodies. That's what makes it polyclonal. All of the B cells from the organism are producing antibodies. A monoclonal antibody is made from only one B cell. So what you do is, in the lab, this is not naturally created in the organism, right? Polyclonal antibodies, everything happened. It was like magic in the rabbit. You just shove it in and then take it out. And monoclonal antibodies, it has to be done in the lab. So you extract a B lymphocyte cell from the organism. So this is the antibody forming cell, the B lymphocyte. And you only want one B lymphocyte, okay? However, B lymphocytes do not live for long outside of the host, outside of the organism. They don't live well in a petri dish. So what scientists actually do, this is really cool, they fuse the B lymphocyte with a tumor cell. The tumor cell, as we know, is really good at surviving, right? They're really good at reproducing and growing out of control. So you wind up getting this fusion of a B lymphocyte and a tumor cell called a hybridoma. You then, you inject the antigen first. I'm sorry, I missed that. You inject the antigen first. So all of the B lymphocyte cells from the mouse or the rat or whatever that is are producing antibodies, but you only want one B lymphocyte. So you extract one B lymphocyte, fuse it with a tumor cell, and then this, what we call hybridoma, replicates out of control. So you get clones of that original B lymphocyte all making the same antibodies. You don't have different antibodies getting made by many B lymphocytes. You have identical antibodies being made from clones of one B lymphocyte. Okay? So these antibodies act like magic bullets on their target molecules. That's what they're called. They're so highly specific because their origin is all from the same B lymphocyte. They're all going to attack the same epitope on the same antigen. Okay? So this is what we more often use for medicine. Polyclonal antibodies, they're cheaper to produce, but they bind to many different places on the antigen because they have, they've all come from different B lymphocytes and they're going to attack different epitopes. A monoclonal antibody is more expensive to produce, but it only binds to one epitope on the antigen. It's much more specific. Okay? So I'm going to stop there for today. Next time we'll talk about an application of this whole concept, the monoclonal antibodies, all that. And it's really, really relevant to what's going on right now. It's called the ELISA test. I can guarantee you that ELISA tests are happening in insane amounts across the country. I am actually fairly certain that ELISA tests are being used as the COVID-19 tests that we're hearing so much about. The swabs are taken to the lab and an ELISA test is run in the lab. So we're going to learn about that next time. So to summarize, today we talked about, the changes in the pharmaceutical industry, kind of starting to rely more and more on the biotech industry. We talked about different types of biologics, right? The ones that mimic cell proteins, monoclonal antibodies, and then chimeric proteins. And then we looked a little bit more about the difference between a monoclonal antibody and a polyclonal antibody and how they're produced in the lab. Okay, so really coming out of today, we've learned how a lot of our medicines are produced. A lot of our more recently discovered and higher quality, higher tech medicines. We're moving away from, just take this chemical to treat your inflammation. Now we're moving towards take this protein to treat your inflammation. So it's a completely different ballgame, something to appreciate. And again, moving forward, you're going to see how this is really relevant to the coronavirus, its treatment, its diagnosis, and its coming up with a vaccine. All right, so thanks for listening. Hope you're staying safe. Take care.